This is collection of all tubs since the filling activity has been completed now. Step 6.1 is instructing the technicians to collect the tubs and place them at the designated table. Record the collection start time and end time in this step. Step 6.2 is instructing you to calculate the yield and take the percentage yield. Record it in the BPR and verifier will verify. This is the same process of calculating the yield on all batch records, no matter if they are for DS or DP or FTP. You see Section 7 on your screen, which is for acceptable quality limit for batch that you are manufacturing. Let's understand what a QL is. As you have already seen that we have taken the utmost precautions to safeguard the drug from any contamination. Now we have filled the DP in the syringes. At this stage, we are performing another precautionary measure for safety, which is AQL. Your company will assign this responsibility to either warehouse person or quality control QC. We want you to understand how this procedure is done, and if you ever get responsibility of a QL, you know the concept and significance of the operation. Every company provides this training to their workers and operators to ensure the drug-filled units does not appear defective, and if it does, it gets rejected at this point. Let's understand with the table on your screen. The first column in this table is the tub number. You know that you have 30 tubs with filled syringes. Every tub will get inspected. Every single syringe will be looked at under the UV light to check if there are any fibers present in filled syringes. That's exactly what your second column is talking about. You would also check these syringes under the UV light one by one to see if there were any leakages. These are mainly two categories of defects that we are looking for at this stage. This training is provided by every company to the operators. The trainer in this training will have you practice dummy syringes with the UV light machines and let you find defective syringes, and you are expected to catch these defects. If you are not able to detect the defective syringes during the training, you will not be qualified for this operation. In this table, operators will check each and every syringe to look for these defects, and if defects are found, you must reject and record under the right reason for rejection. Moreover, calculate all rejected syringes, if any, and final number of syringes and tubs will be stored in right storage condition. Let's move to those steps before completion of this training. Step 8.1 is instructing to affix the labels appropriately to each and every tub. Step 8.2 is to now store and place the labeled and filled tubs to the fridge under specified storage conditions. Don't forget to fill the fridge log here, and this log must mention which drug lot is stored, under which name and strength. All these details are mentioned to create more and more traceability. Now you have successfully completed the manufacturing activity. The rest of the BPR is the same across the globe. Just like the DS batch that you learnt last, the last page is for deviation and BPR version update and history, which we will see next. Now look at this page. This is usually one of the last few pages of the BPR. You have learned about deviations in terminology class as well. Again, for your reminder, anything that doesn't go as intended during the manufacturing process is a deviation that must be recorded here with its brief description and conveyed to the supervisor as well. That is all. The very bottom section is for the history of the BPR. Anytime if there is an update to the BPR, even if a spelling mistake correction from when the BPR was created first. Every time something is changed, another version will be created, and the reason for the change will be mentioned here in this BPR. This is part of traceability, which is required by the FDA. We have discussed the traceability part over and over again. Now you have successfully completed your second assignment and know the DP process as well, with simply using formulation recipe with DS and excipients. If you get a chance later to work for a company that manufactures DP only, you know right of the bet that you will be working with a process similar to this. The drug recipe can be different, but the formulation would be same principles. DS will get mixed with excipients and binders under certain temperature conditions and mixing hours. Whichever is more critical to the process, but all these instructions will be provided to you step-by-step instructions. Their temperature, agitation speed, timing, in-process samples, gowning practices, buffer preparation, pH readings, sample submissions and logs, your understanding of who will issue the BPR, where you would go next to receive the materials, who would sign which log, is the information you need to understand and learn to become a great technician. As long as you have understood the GDP and GMP principles, understand the importance of filling logs, creating full traceability, following training compliance, not executing on anything that you are not trained on, make sure to make a practice to always check calibration and cleaning tags of equipments before using them for drug safety. These are the very common mistakes that happen day in and day out in our industry. If you know and understand and make these practices a part of your professional profile, then you are already ahead of the game in your profession and a valuable technician that a company would like to keep. See you in the next training, where we will take this DP and will turn that into a finished drug product. Yes, FDP. The only thing that will be added to these syringes is the labels and primary and secondary packaging. That is an interesting topic and a final one for you to learn before you can go out in the real world and say, I am ready for this job. See you there.